Yeah, so I think, Glenn, we can start, or I will start with short introduction,
Yes.
and then when Lucas will join us, he will just continue with today's lecture. So, welcome everyone. Thanks for joining our third FunDive webinar. And today we have Lukáš Picek. I think currently he is stayed in MIT in the United States with the Fulbright scholar for postdocs, I think, but he will introduce himself for sure more in the details. I know he did a PhD in Czechia, in Czech Republic, and he will be talking today about his main expertise, which is AI, I think machine learning a bit as well, and stuff like that. So, Lucas, I think we have already a time to start, but maybe Glenn, 1 minute, quick reminder about our rules, and then we can move on with the seminar.
Yes, thank you, Marta. So this is our third webinar. Welcome, everybody. The rules are quite the same as the last time and the time before that. So right now you're all muted. You will be given the opportunity after Lucas has finished his talk to put on your cameras and to unmute yourselves. If you have questions in the meantime or you think of anything during Lucas's talk, you can just put them in the chat. Marta or I will read them out loud afterwards so that they're also on the recording. And then hopefully Lucas will be able to answer what you want to know. And with that, I would like to give Lucas the opportunity to share his screen.
Cool. So very nice to be here. Thank you for the introduction. And let's try to share. Oh, okay. So not this one. So overall, wait, I don't see you, which is quite
This is how it is.
unfortunate.
Yeah,
Okay.
unfortunately. Yeah. But I think it's
Okay.
it's normal on Teams, yeah.
Okay, interesting. Anyway, I'll try to I'll try to speak to myself and potentially have some questions later within the webinar. The webinar is split, or let's say I split it into three sections. The first one is more about practices and lessons learned. It's not about like a general take on AI for biodiversity. It's more or less my take, and it's my outputs or outcomes I've worked on with other people, more or less. And the second part supposed to be about demoing an app, and the third one is more about the technical aspect. So for those that don't want to stay for the technical stuff, they can definitely leave, or if there is no time, we can skip the last part, so on. But anyway, thanks for having me once again, and let's, without further ado, let's just jump into that. So I'll start with a very simple question, and that's what is the species identification? Because one of the goals in biodiversity ecosystems is to know what is the species and so on. This is a very sensitive topic in biology in general, but I will give you again my take on what it is and how I, as a machine learning guy, approach that. So for me, if I'm given a specimen, the species identification is either about giving it some kind of a common and scientific name, which then from machine learning perspective translates to something like we have to find some kind of a label from a pre-selected set of labels. I mean, this seems unnatural, but this is what majority of algorithms do or work on. And despite the natural obligation is a little bit different, and there is a lot of species that are not discovered yet and so on, That's one of the pitfalls of the species identification and itself in general. Despite we can very narrow the scope, so it's like one species from the set, it is super challenging due to many reasons. One of the reasons is that there is a lot of species and they look the same, and they don't look the same if they're supposed to look the same. So given the 5 examples I showed on the slide, for the first row, this is the same species, but you can see the variety is pretty big one. For the second row, it's actually 3 species, not 5 different species, but 3. And it's very hard for even a botanist to say which one are the same. Similarly, a lot of species develop to look alike different species to protect themselves for predators and so on. This is pretty common for insects or snakes. Not that common in fungi, but some kind of morphology that is definitely a case for fungi could help or actually worsen the prediction accuracy and so on. The next part is it is hard due to in the wild nature of the observation of the species. So if you look at like the constrained environment where you can photograph or scan different organs of a species, it's usually solves the problem. But that's not how it usually works or what we are actually looking for. So usually you could have like different way how the species looks like through the whole year and different periods of time. There are other species occluding the species of interest. You have different parts and so on and so on. Then we have something which we called in machine learning a domain shift. So despite on, let's say, a benchmark or set pre-selected before development, we could have a super nice performance. And when we try to deploy it in the real life, let's say, you usually encounter weird observations that you never encountered before, which results in, let's say, worsen of the performance. But usually, like, you cannot always get the sense of what the people will submit in the future. So it's kind of a long story to tackle those kind of challenges. Last but not least, it seems we do have a lot of data. We do have a lot of opportunistic observations. There are billions of data points in GBIF and millions of species. However, they are strongly biased towards the Northern Hemisphere, or especially, North America and Europe, where the global species distribution is actually centered around the equator. So it doesn't tell us much, but also we can still use or have some use of the data. So what could be the perspectives, like where we could actually use some kind of machine learning or AI and species identification itself? I would say there is much more than I put on a slide, but in general, there is a lot of use for ecology and conservation. One particular example from my experience is, to prevent snakebite mortality prevention, because in many neglected countries, and developing countries, there is a lot of issues caused by snake bites. Namely, it's hundreds of thousands of people dying a year because of not having the right antivenom and so on. We've also have field guides, so you probably know a lot of citizen science applications where such models can help people to learn and also, engage more with nature. There's also a huge possibility in agriculture and possible in pharmacy as well. Which brings us to how we can use, or how we used, and how we can benefit from using machine learning in mycology. And this part I will tell as a story, because it is something that is evolving for currently like 8 years. And it all started with Jacob organizing a competition for machine learning folks that I turned out to won at the end. And that resulted then in our collaboration, which now spans like eight years. So what I would say is an interesting perspective is how we can use the citizen science to help us collect more data. So the old school data collection you probably are familiar with is you have to go there, you have to be there in very uncomfortable conditions and weathers, and really observe the nature. So you probably know that this is very hard to scale because there is not many skilled people to do that. And also, you have to collect, you have to sample, DNA sequence and so on, a lot of data. And, it's hard and it's time consuming and it's expensive, right? The other take or other idea is, okay, let's go with social media. Those are, by the way, 2 posts from Czech Mycological Society group for fungi recognition on Facebook. And I'll just translate briefly what it says. The left post is, I just ate this and I don't feel good. And it also, my 9-year-old son ate that as well. Are we okay? Is it poisonous? Is it okay to eat it? And the other one is very similar. We ate those frozen mushrooms and could be poisonous. And basically, this is what you have to deal with if you are on a social media. And you also have to deal with a lot of, people, which means there is a ton of spam, there is a ton of, private messages with some swear words and so on if you ban people for not, following the rules and so on. And that, by the way, is a good way to collect the data, but you have to always deal with the negative side of that, and that's you have to deal with the data. What is a different way, which I think is a pretty good one and works pretty well for Denmark, is let's have a relatively small group of enthusiasts to collect the data partially, and potentially also sample data for you and send it if it's possible. So overall, you probably, and I hope you heard about the app that Jacob and Thomas and other people from Denmark developed back in, I think, 2016 and maybe 14. I don't remember, sorry. Which in general is super simple. It is, available for both platforms. And the only thing it does is you submit a couple photos, you submit some details about habitat, substrate, and so on, and it gives you 10 suggestions. It's ranked list of most likely to be this species according to your data. And it doesn't show any confidence. It doesn't show anything about edibility. But it just, gives you some rough idea about what the species might be. You can also add some comments. You can discuss further in the process with the mycologist, because if it's an interesting specimen, you can get some engagement with the mycologist. And in general, it's something that helps to scale the sampling a little bit. For those that don't know how it works, you just, select your photos, or photo your photos, or the mushrooms, your specimens, and then you get pretty fast some kind of a feedback and possible prediction. Okay, let's see. Okay, sorry. Okay, so how about the performance? I mentioned some possibility of being correct and incorrect and so on. So when we started back in 2018, we used, let's say, something like a state-of-the-art machine learning approach. We used some deep neural network, the state-of-the-art architecture back in the day, and we reached something of like 70% accuracy. That means that the top one listed species was correct in around 70% or 70% cases, where in the top 5, it was usually present because there was like 9 out of 10 cases, it was there. That's, by the way, super interesting, because if you then plug a human into the loop and present him a top 10 possibilities and ask him to select which one is the most likely to be correct one, you can get a pretty big performance already. So this integration of let's ask human based on some, selection and verification of different images to select the right species, you can get pretty high accuracy overall. So speaking about the performance, that's something that I would say is not important at all, because people usually don't or cannot like analyze or understand the accuracies and the way how we as AI community interpret the results. But what I would say is interesting from mycology perspective is that just introducing the system that can help you to observe and learn, it skyrocketed the number of observations and active contributors per year. I mean, despite it looks like a small number, like 1,600, it is a lot of people that are engaged on a monthly basis, and they submit every month couple observations and so on. So they are the most valuable, I would say, recurring users that submits a lot of observations from, let's say, similar locations across years, which I think it's the most important part. We also, let's say, doubled the number of red listed observations, which I would say is the important part. And as you might know, and as Jacob published that back in like 2018-19, couple species that were considered extinct in Denmark were rediscovered because of the public interest in the observation of fungi. What is also interesting is if you observe the observers, you can find that experts or taxonomic experts usually tend to focus on their group. So the graph I show on the right side displays different types of users, different types of groups of users based on what they submit. And in this case, it's submitted into eNaturalist. And the first line, there is, by the way, no cursor, so I'm sorry that I cannot show it, but the first line describes or includes 60% of the users, and those are the non-experts. And you can see that the diversity of the different classes is much bigger than if you look at the domain experts. And you can see it kind of reflects the bias the domain experts is working under. And it's like, yeah, if I like this particular, genus or something like that, or class, I will usually submit a repetitive observations of this particular species or, species within this class. Whereas the non-experts, they usually tend to learn. So like if they submit, let's say Amanita muscaria, they then learn that it's Amanita muscaria, and they usually don't submit or resubmit similar species in the near future because they've already learned, and they don't look at it as I'm observing the nature and submitting everything I see. It's more about the learning perspective. Overall, through the use of the app, not just through the use of the app, but given the app, the Danish Mycological Society collected and reported in GBIF around 1.2 million occurrences, which are a subset which is, let's say, curated and pre-selected to have some quality. That means labels are more or less correct. There is GPS location. There is a lot of metadata and habitat information, and so on. Also, there is something around 250 studies that use the data and form some kind of research outcomes based on the data, which I think is also important, and it tells us something about the importance of sharing the data and not keeping it for myself, but also sharing it with our peers, because that's, from my perspective, the important part as well. So if it helps mycology, like why I should engage as a machine learning guy with the data and with the community. So I believe the data itself, since it's well curated and it's, a lot of different observations of very lookalike species, I think it can be, and I still believe, and I hope it will be recognized more, but I believe it is a form for evaluating future machine learning models. And with that in mind, we published or created a dataset for machine learning guys, which is a subset of the Danish, data from Danish Mycological Society, including something like 1,600 species and 300,000 images. But majority of them have location, habitat, substrate, and so on. And despite there is like a very long tail distribution, and you have, a ton of species with just few samples, it is a good test bed for, future development of machine learning or AI algorithms. And that's also something that helped us to build a better model. So basically, just 2 years later from, original model, original implementation, and real field, use, we get something like 15% improvement in the top one, which interestingly is not that reflected with the human in the loop performance, because the human in the loop performance, despite we lowered a lot the error of the model on the top one prediction, did not improve that much. So we went from, let's say, 92.8 to 95.3% accuracy in terms of people selecting correctly the species. In other words, 5% of the samples were labeled wrongly. We don't count those that were not selected and so on. So I would still say just 5 samples out of 100 wrongly selected by human is actually pretty accurate. And it is actually pretty close to human expert performance, because given you are a mycologist, from my perspective, you are an expert, but I know you are, or people are usually just domain experts and they know, I don't know, Amanitas and Rusulas, something like that. However, given that, you can actually, be wrong in 5 cases out of 100 for the taxa you don't know. And in general, with more submissions people tend to submit, more accurate they are in their submission. So I would say that brings us back to the original idea of people actually learning by submitting more and more and more. The other aspect that is and was super interesting for us while forming the the Danish fungi dataset was to measure how the models can and actually do misclassify the toxic species for the edible one, or the poisonous for the edible one. And interestingly, since majority of the species is it's edible, let's say, not poisonous, the model learns that it's easier to mistake the edible for, or poisonous for edible, because there's not many poisonous species, right? So it's the prior probability that that just makes the problem. So interestingly, despite you have something like 5% accurate, 75% top 1 accuracy, 1 out of 10 samples that are poisonous are actually classified as edible, which I would say it's a major issue, right? If you look at how the edible species are confused for the toxic ones, and also it's super interesting to study how, from machine learning perspective, obviously, it's interesting to study how models can actually learn and work with such a meta information in the process in order to have at the end same performance, but let's say reflect the poison species to edible species confusion much worse than the other way around. Because I think we all should agree that confusing the poisonous species for the, let's say, edible one, and people then eating the mushroom is making some kind of harm that you don't want to do. So with that in mind, we formed another iteration of a machine learning data set, which we released last year, and it's supposed to account for all the future problems, and it's actually a present problems as well, where we deal with many classes having very few samples, and there is a lot of species coming into the system, or observations of species coming into the system that we have not trained on. So our idea is to make such kind of recognition better in the future, and we therefore add different subsets and a lot of different metadata, including captions and segmentation masks, for the future training and potentially use for mycology practice as well. So that's, let's say, the first part, where I would say there is still a lot to do, because we haven't touched the data much, I would say. And I would like to showcase like where we can move potentially with the fungi, and where are we basically with the similar approaches and similar models in botany or ecology of vascular plants. So you probably heard about a PlantNet. I'm not sure if it was said in the introduction, but I spent past like 2.5 years as a part of the PlantNet team, where I was working on, different ways how to use the PlantNet data. And for those that don't know what the PlantNet is, it's an citizen science app platform that helps people identify plants using a mobile phone. So it's very similar to the Danish app. It's just much bigger. You have 30 million users. There is something like 2 billion observations in total. We have data from 200 different countries, and we are also one of the biggest contributors to GBIF. The use is, let's say, pretty wide. On the biggest day so far, by the way, we've measured around 2 million observations or identifications a day, which is still unbelievable. It's super big number in general, but the usage spans both the personal and professional use, and we cover more or less everything. What is, I would say, still an issue with the data we have is that despite we cover all the countries and so on, the data are extremely sparse on the species level. So if you look at the right map, let's say there is like 50 to 100 kilometers from the Montpellier to the next points of reports for that particular species, which doesn't tell you much about like local presence of those species. Anyway, despite the data are sparse, we can use them somehow, and our goal was to make something which we call now a GeoPlantNet, which is a platform for displaying maps of thousands and thousands of species and different habitats and different I think I'm missing one slide, but and different biodiversity predictors and so on. Before I'll do some quick demo of the GeoPlantNet, do you have any questions so far? I think we can probably jump straight to the questions because we have like 30 minutes since we started.
I mean, we can probably, if you don't mind, Lukas, we can wait with the questions until the end, or if someone wants to ask now, then you can, but we can also continue and then people can post them in the chat, or I think it's up to you what you prefer.
I don't mind like having questions now. I know you said the rules are like that, but since we will then jump into more, let's say, technical stuff, I think it could actually be a little bit convenient to maybe have the questions here.
So then I can have a question. So if you don't mind, I can ask because I was thinking, like, I have two small questions. One is they are very simple because I'm not experienced in that things at all. But I was wondering if mushrooms or like fungi in general are more tricky than comparing to plants or, for example, insects to be used for this sort of algorithms for the identification. Is there like some extra challenge within mushrooms, or it's not the most tricky group to use in sort of thing?
Yeah, it's a good question. I would say it's one of the most trickiest. I think corals are the most trickiest, but fungi itself is super hard. I think there could be even a couple people in the chat just questioning what I said, because many people think you cannot distinguish any species by just visual perspective, or even having some kind of meta information, and you always need some DNA sequencing and so on. I partially agree, because yes, it makes sense. Maybe all of my predictions are wrong, but they are wrong from your current view on the species, right? And what is the species, and what is your, taxonomy, and so on. So yes, it is probably wrong, but to your present knowledge about the species itself, it's the most correct way. And for many species, yes, it's super, super easy to interpret the results. And I mean, even having like 2-3 visually similar species in the like ranked list is super helpful for people, because they can understand that there are similar species and they should be aware of there are different species that could actually be harmful or whatever. So I guess it is super hard. And I still, despite I showed you like, yeah, top one accuracy is 85%. This is far, far from the reality. Like for real, it's probably much, much, much lower. And we are still talking about, let's say, 6,000 species, right? We are not talking about the rest. And it's still only the, those species you can see above the ground, basically. So long answer for your simple question.
Yeah, but I totally understand, and I see a lot of potential, and there will always be these discussions like what is the species, that's first of all, and then yeah, about how accurate is like whatever researcher and whatever person experience level and so on, and so all of those things counts. Also, what I was wondering, if because in this like sort of documenting of biodiversity, there's like millions of pictures with the surroundings, and a lot of photos are not providing all of the morphological features that are like straight away immediately visible on the photos. And I was wondering if, for example, algorithms can be trained or like designed in a way that they can use this extra information on the habitat that is surrounding as an extra morphology or like extra identification feature. Like, for example, this is a mushroom that we don't really see specifically, let's say, structure of the gills or something like that, but we see that it's specifically on pine tree. Is this like things like that are included in these algorithms that are identifying, or it's like not so, like it's more probably complex, but it's like I don't know which things could count in the general, let's say, identification support.
Lucas, you're still muted.
You're muted.
Sorry. Good, good catch. So if I'm being specifically about the Danish app, the information about the habitat and substrate is used. But you can also use a ton of different meta information, which also leads to some kind of biases, because I assume you also want to know about the rare cases where the species occur where it's not supposed to be. I think those are the cases that you can actually kind of disregard using the site information as a prior probability. So I hope it makes sense, but imagine you care about like animal movement, right? Like imagine you care about, I don't know, wolves or mooses or something like that. And you never seen a wolf in your area, and now they start appearing in your area. So you need to know about that, right? You need to know. But your prior is set for there is nothing in here. There are no wolves, there are no bears, right? So you need the models or the algorithms to account for this one as well, which also leads the con for not being always correct. One interesting point is that the way how people observe nature and, let's say, fungi species is completely different from the algorithm. The algorithm is just statistical, perception of what is in the images, right? So, and in the metadata. So what sometimes is hard to imagine, like for people, is why is this prediction as is? And the answer usually is like, yeah, this is statistical significant. That's why. That's why. And that's the only reason. And usually this works in, I don't know, medicine, where, the people can, follow the intuition, right? Like, and the great surgeon just do the decision because it's like gut feeling, right? I have to do this because he did it a million times before, and he's probably good because he did it a million times before. And this is the same with the models and the machine learning and so on. So it will be good. You probably always, like, you all use some kind of GPT or like, chatbots these days. So you know that it is kind of good in the general stuff, but more you go to the like edge cases, it's getting worse and worse and worse, which brings me back to, yeah, that's why we build like the third or second iteration of the data set for machine learning, to kind of encourage people to tackle this from perspective of hard species, hard problem, and very, very limited amount of, resources.
Yeah, thanks a lot. Yeah, it's actually yeah, it's now a bit more clear in my head. So thanks a lot for your first part of talk and for your answers to my questions. And I will just leave the floor for some other people maybe asking questions, or we can continue. I will adjust with the preferences.
Seems no other questions? Or Glenn is talking,
Glenn,
but
if
he's
you are
muted.
talking to us, then you're muted this time.
Yeah, I just wanted to say that you all have the ability now to unmute yourself and show your video. So if it's on by default, you'll now be visible to Lucas. I maybe also have actually I do have a question for you, Lucas, before you delve into the technical stuff. In the beginning, you said that the machine learning, the way it works, is that you have a predefined set of labels, and that from that predefined set, then the machine learning ultimately has to select one. And my question is, I guess, related to those of Marta, is if you have an undescribed species that forms a visual cluster that you can, in theory, visually recognize, is there a way to kind of label this as like it's not in the data set, but we've seen 100,000 images of this, and this is probably something distinct?
Yeah. Super good question, and it's the way how it's evolving these days. So we are getting into. I think I have a slide about it. So basically, this cluster I showed here. So if you look at the different colors, it's the, let's say, a 2D visualization of the image, and different colors means different genera, where the different little clusters are usually a different species. So if you train the model to distinguish the species from this spatial perspective, you can then introduce a new species by providing the samples and probably landing somewhere here or here if it's within the particular genus, right? And this is something we still explore, but it's definitely the way forward to allow such kind of, future introduction of a new species into the model, because it is something that bothers us a lot. I think I measured that a couple weeks ago. Around like 20 to 30% of the submissions to the app are species not trained, or like species that the model was not trained on. Like, it's so many species that, and like there is so many new species always added into the database from different countries and different perspectives that just we don't have, or we had not access before. So that's how it's evolving.
Yeah, cool. I saw in the beginning that a lot of the for fungi at least, it seems that on GBIF the world is in Europe and North America it lights up quite well, but for other parts of the world, of course, it doesn't. And what was a surprise to me is that India has quite a lot of observations, and I can imagine that there's a lot of species to identify in this way in that part of the world.
Yeah, definitely. The question is how to encourage the people. I think the way of like the reason why there is so many observations in North America and Europe is like we have time for that, right? We don't have to spend 16 hours working to, keep ourselves alive. We have this, precedence for, we have a good salaries and we live in completely different world, and we have the time to actually go to the nature and just observe, where the majority of the world don't. So I mean that's what it is, but we can see probably in couple 10-20 years that they will develop into similar similar state as we are currently. And with more and more data coming from the region, I think we can also account for, or like we can just point it back to this social or economic impact, or reasons for not having the record, like the samples from there.
Yeah, thanks. That was quite clear. I think we should ask if there's any more questions by now, and if not, then you can, I guess, continue.
I don't see any raised hands or whatever. Okay, so I'll just move forward with the GeoPl. So I promised some kind of a demo. So I think what would be helpful for the fungi community is actually to build something similar we built with the PlantNet team on the GeoPl. And basically, we want to map all the species we have, sightings about, and we want to predict some, biodiversity hotspots. We want to predict different locations that might be interesting or have some, common ground with other locations and could host, let's say, some rare species and so on. So that's something we did for GeoPl, but has not been done or discovered or even potentially discussed for fungi. So I'll do a very short showcase on a friends. So it's available for all the European countries. So you can play with the app as you like. It's available on geo.plantnet.org. And the overall idea is if you select a country, you have some global report on left with some highlights about red listed or endangered species, different habitats, different tree and invasive species, and so on and so on. You can also go into like separated or a species list and select your species of interest and explore where it is. What we can do is check, for example, the critically endangered species and check their presence. So this is just for your information, a species that should occur around the rivers, and they usually do. Interestingly enough, there is I think there are only 3 observations in the whole France that results in this extrapolation. But if you actually verify if the species is present here, it's correct, because there is much more of the observations in Germany on the same river. So that's one of the interesting facts, that we don't interpolate the observations recorded, but we try to extrapolate on different biological indicators, predictors, let's say. What we can also check is like, different habitats. So if we go for like, I don't know, Mediterranean, let's see, Mediterranean, yeah, let's try this one. I actually like Mediterranean Sea or something like that. It's like N14. Yeah. So it's like Black Sea, Mediterranean, coastal dune habitat. We are following the UNIS habitat mapping, which again could be could be much different. But if we go and see the different predictions, you go into a pretty good precision on, okay, let's check the satellite, on the habitat of like coastal dunes at the Mediterranean Sea. This, by the way, what is available is a version 1. We have a version 2, which we currently evaluate with botanists from many different countries and providers of the data. But in general, I would say it's quite precise already, even for the version 1 itself. So if I go to, let's say, indicators as well, one of the points that could potentially be used or have some impact is the species richness and potential selection of locations that are interesting enough for whatever reason you have, introduction of reintroduction of species, for example, or just discovery of what is there. If we check the species richness, you can see some kind of hotspots here and there already. And what I like to show is actually here you can see that there is an airport, right? So like when I first saw this one, I was like, this is so weird. Like, why there is a biodiversity hotspot next to the airport, right? It's so strange. But if you just switch to an OpenStreetMap, you see straight away, like there is a natural reserve. And it actually makes sense that there is a biodiversity hotspot at that location, which is more or less the same for, here are another heat spots. So if you look, there is again a natural reserve. If we move a little bit up, it is also highlighting a natural reserve. So that's something I would say backs up the validity of the maps itself. And one of the potential uses that I actually like is if you go into the city planner, you can always have some report for that particular location. So if you are some politician, local developer or whatever, you can use it for planning of the city expand and so on. So for example, if you draw some kind of a polygon here, you will realize sooner or later that there is a vulnerable species at that location, and you should probably move somewhere else, probably somewhere like here, where I hope there is no vulnerable species. Yeah, so polygon 1, yeah, there is some vulnerable species, and in general, there's like 80 different species. For this one, there is no red listed or vulnerable species. We can also just check for that species right here. Let's see. Yeah, it's loading. I hope you can still hear me and it's not my internet.
We still hear you, Lukáš.
Awesome, awesome. So I just like the app. But in general, this is something that I was about to showcase, where on the European level we can display, and predict different habitats, different biodiversity indicators, and so on and so on, just using the observations and a lot of different predictors, which is the technical part I will dive into a little bit. And since it's very similar, I think we can, or it could be pretty easily used or reused one-on-one to map like compositions of species across taxa, and also across countries and so on and so on. So I'll just jump into the third part of the presentation, which is the more technical one. So we've seen the GeoPl, which is actually work of many people. So within the PlantNet, there is a lot of domain experts. There is a lot of just like core developers for front end and back end, and then there is like a lot of researchers, usually from machine learning perspective, but there is a lot of ecologists and botanists as well. So our goal in general, we want to, have some general information about the species compositions and distributions, and to monitor some ecosystem health on a larger scale and for a longer period of time. So our idea is that if we predict the map every, let's say, 3 months, we can then sooner or later see some shifts in the ecosystem degradations or so on. Very shortly, how the whole pipeline looks like. So we take a ton and ton of observations from European Vegetation Archive and from the GBIF, where majority of the GBIF data are the PlantNet observations. We take a lot of climatic and satellite time series, and a lot of different predictors. Using that, we train a deep species distribution model, which is convolutional neural network that allows us to directly output the species presence for more than 10,000 species, and also then convert those into biodiversity indicators and species distribution maps. Following that, we do have the information about the presence and absence, or pseudo presence and absence of the species. We can then model some habitats model, or train and use habitat distribution model for modeling different habitats. In our case, is the UNIS, different levels of UNIS habitats mapping. The data itself, as I said, includes a ton of stuff. And what is interesting is the right part is all the opportunistic data submitted by people, where the left part is the presence absence data submitted by the botanist. So imagine that's like the 1 meter square box put on the ground, and then someone spends a whole day like recording everything that is present and absent, and repeats that in spring, summer and autumn and winter. So you really have some sense of what is present at that location. As I said already, despite there is many occurrences, for some species it's very sparse, which is an issue. I think you might have some idea about how this is evaluated. We usually do something which is called spatial block holdout. So you don't want to have same locations for testing of your model. So what we do is, so instead of like having random samples where you would have similar conditions for both training and validation, and you would like overfit to those conditions, you do something like, yeah, as I said, spatial block holdout. So you have grid and you randomly select from that grid into your training and validation, and you repeat that multiple times. So you have different training and different validation sets over and over again, and then you just average that on your performance. So despite there is some kind of a leakage, since we sample multiple times randomly, we can reduce the bias, or the sense or impact of the bias on the measured performance. We also have to account for the biases and fine tuning to the biases and out of distribution test sets, which is the yellow points on the right side. So it's completely distinct, different countries, different regions, and it's actually just to test much better how we can actually extrapolate in locations where we have literally like very limited amount of data. As I said, we had a lot of different predictors. So we have something like 40 years of time series climatic data from CHILSA. We have something like 25 years of satellite data from the Lancet satellite. And we do have a ton of environmental rasters, which we interpret like a single value for each of the locations. And for each of the locations, we also have relatively high resolution satellite images, which originates from Sentinel-2, and they have a resolution of 10 meters. So for each of the locations that we have in our data, we extract the multiple bands from the satellite at the 10-meter resolution, and we add some context around, so you have an idea if it's near a field, if it's near a coast, if it's near a city, and so on, which I would say is an interesting information for the prediction itself. We form the time series data into the cubes, where we have different variables across years and quarters. And for the Landsat and for the climatic data, what I think is super interesting is that if you look at the climatic variables, despite there are differences, there are not much differences in Europe, like in general, right? Like the climate more or less similar, so there's not much of a difference. So if I tell you those are same days in Switzerland, France, Germany, and Denmark, there's not much a difference in how it looks like. I mean, there are obviously a lot of differences, but it's not that big as if you look at the visual perspective or visual domain. So same locations, just the satellite bands or satellite information, you can clearly see that this one is much different from visual perspective than the other, and vice versa. So a little bit more into how the models work and what they do. So since the Europe, if we divide it into 50 meters times 50 meters grids, we need something that process 5 billion of those grid cells pretty fast. So in our case, we aim for training a model which is compact, and therefore we used a very, very shallow architecture, which is in-house built, and consists of 3 modality backbones where we feed different modalities into different encoders, then we concatenate them and then predict the species assemblages from them straightforward. Those are then used by the deep habitat model, which consists of a large language model that is, I would say, pretty similar to what you can encounter in ChatGPT or any other chatbot, where the goal is to train a model based on something like removing randomly the inputs to predict what it is, which habitat it is. I don't want to tell you more detailed stuff, but in general, how it works is if you take all of those, if you take for each of the points your labels recorded by the expert, and you extract your multimodal inputs, you can then train the model, the species distribution model, to predict what are the labels provided by the expert, despite you don't show the models straight to the model, but you use it more like model the error than using it straight as the model input. Yeah, I know this is kind of misleading, but since this is used as a stuff for model the the problems, I think it's quite okay. So given that, you can then, for each of the plot, take the SDM output as the species names, which is the language, and you interpret them as the tokens, and then you more or less use those tokens as to predict the species, or not the species, but at the end, the habitat based on the species. Within those steps, there is something which is called usually a dropout. So you drop randomly some of the species. So you model, accounts for, not everything is predicted. And that's why I call it in the first place, it's a pseudo presence absence. So it's not a real presence absence observation, but it's some kind of prediction, which can actually miss some of the species. And you want to model something like, what is the species composition and how the species composition is aligned with what is supposed to be within this habitat. And again, I know what is the habitat and biotope and whatever is super sensitive topic in biology, but we follow what we were told and do what we think it's important to do, and like distance ourselves from the biological issues and the way how people from biology look at the definition of the problem, and we just take one and do it as we think is the good way, or what is the good way from machine learning perspective to do it. But in general, we can always apply it to any other definitions or nomenclatures or taxonomics or whatever. So that's it. I don't think we have to go more into the performance itself, but in general, the performance is quite good. We try some standard stuff like XGBoost and Maxent. Maxent is like a standard for species distribution modeling in general, but since we have so many species, it just failed. Like, it's not possible to model it. It's just too hard. Multiple people tried it. There's still a chance that it actually, there's something wrong or we do something wrong, but I still believe it's just the complexity of the data that makes it impossible for the Maxent to model the distributions for so many species at the same time. So our model at the end, if you compare it directly with the XGBoost, we got pretty big improvements using only the single modalities. But if we use the whole ensemble of the different modalities and different encoders, we can go much higher into like sample average F1 of like 33.6%, which is super big, considering there is 15,000 species in the test set. The out of distribution performance, however, is super bad, which is expected, and it's something we are looking into for the past 2 years. So I mentioned we do have some test set, that's the yellow, which is nearby all the observations we have. And then we have a complete test set, which is very far from all the observations. And if we just look at the numbers for one of our competitions we hosted, we can see a difference in performance by, I don't know how, like people achieving 40% in one of the benchmarks or competitions then drop to like half of the performance, or I don't know, 70% of the original performance, which makes sense because the data are so different and so bad that it actually makes sense that it's not gonna perform well. But still, on the other hand, the 23, sample average F1 score on like never seen or countries where we have no data from is super great, and we are just trying to lower the difference between the out of distribution test set and the in distribution test set. Which brings me more or less to the end of the talk. I obviously have millions of different slides and different things to add, but I try to be as less technical as possible and try to, give you some insights on what is possible in plant biology or botany, and kind of give the perspectives for where we could go with the fungi, because I believe it's possible. It's just something that needs a lot of time and discussion probably before, and so on.
So probably I can say thanks a lot, right? Is this the end, or it's
Yes, it's the end.
Yeah, so thanks a lot. It was super interesting. I really enjoyed. I learned a lot, and I think I can start the discussion. We still have some time, and people that did not manage to ask earlier, they can ask you about the first part of your talk, or is there any question? You can, yeah, because Glenn allows you to use your microphones, and you can also turn on camera if you want, or you can post the question in the chat. So I can give you some time, and I can actually start because I have a small question. It's like I'm totally out of the topic, so I'm not an expert at all. But I was thinking whether, because it sounds for me like I compare this situation to this like genomic data used for phylogenetic analysis. It's like not always more data, or like genomic data is supporting finding the best hypothesis or making our output less complicated. And it sounds like that when you have small scale, let's say not so complex parameters that you use for your modeling, then it's like a bit simpler. But then when you concatenate them in the bigger clusters, let's say, it's then more tricky. Like you said, that this one pipeline didn't work as you expected, right? So but at some turns it worked for something, so it's very similarly tricky, I would say.
Yeah, definitely. And usually people use it on, I don't know, in US, like California scale, right? So that's compared to Europe much smaller. Or pretty commonly they use it in Switzerland or Denmark, right? Like locations where you have ton of data and they are compact, and they're like, Denmark is like not many biotopes, right? Denmark is like it's so easy, kind of, right? So that's definitely a challenge, but I think our goal is the extrapolation itself. So you potentially discover some hotspots in regions that are under-recorded. So for the plants, it's usually the Balkans. Like the Balkans are not reporting anything. So using this as a tool to identify the hotspot areas and then go there, discovering is like you're aiming to something which makes sense. And we do have some feedback already from a couple botanists, and they're like, yeah, like you are predicting a lot of species that are not present in, I don't know, Czech Republic, for example. And I'm like, are you sure? Like, can you put your hand in a fire for like being 100% sure it's not there? Right? Because that's the point, that there is some knowledge about what is the species distribution, but that's probably wrong, right? And I know it actually opens a very serious discussion about this is super interesting for reintroduction of the species, right? Like they were here, you can find some hotspots that are, I don't know, far away from, people, to make it easy. And you can just plant the trees there and it would probably prosper in the future. And there's many points to take it, but in general, yes, it is complicated. And yeah, that's it.
Yeah, thanks a lot for the answer. I know it sounds very tricky in general, but I see that there is a question from Vashko. So Vashko, you can ask if you want. Hello, by the way.
Hello everyone. Hello Lucas. Thank you very much
Hey.
for your talk. Very interesting. I wanted to take the conversation a little bit towards the validation of ground truth, and I think you kind of touched it just now in your previous answer. Because experts, the word expert, it can mean many things, especially in fungi and mycology. You can have experts that can be wrong half of the times, and even very top level experts can be wrong at least like 10 or 5% of the times. And assuming that the validation of the ground truth can only be achieved with DNA, let's assume that, and assuming that the top expert can make a mistake of 5 to 10%, I would like you to comment on the risks or the implications that could have in the quality of potential tools.
Yeah, I think we touched this question already. Like when I first started discussing anything related to fungi, I thought that DNA sampling equals 100% correct labels, but then I was laughed on that this is not correct at all. So even the DNA sampling could be wrong. And it's related to how well is the bank, formed, and how the algorithm to match the DNA samples work, and so on. So you're always pretty far from being perfect. But in general, I can have two takes on it. The first one is I don't care. That's like the machine learning point, right? I just want to have a better model and I don't care, right? Which is not correct from your perspective, right? Because I will just tell you the model is better, it's improving, you're using this and this and that, and it's going to be better and better and better, which then just brings us back to your question, which is, what is actually correct, and is 95% okay and not? And I don't have the answer for that. Like, I think I think 95% is good enough. And if the best experts cannot give you anything better, there is not even a way how you would evaluate that. Or do you think there is a way to do it? Because right, if there is nothing like a better truth, what is the truth then, right?
Well, I think it's pretty consensual. I think most people would agree here that the DNA is more informative and more accurate as long as it's well curated and well analyzed, because you have to be some sort of an expert also to analyze the DNA, right? So it's just like the DNA by itself. It's just like anything else. It's just a character, right? But it varies much less than the color of the gill, or and so on. So it's a little bit less variable, and it can be more informative if well analyzed. So
yes,
so I'm just wondering if because that's also hard to get, right? Like it costs and it takes time and so on. But if we have a tool that doesn't differentiate species A from species B because there was no ground truth, or the best ground truth was the human, and here we are assuming that the human is pretty good, but just indulge me. Let's just, maybe it's not really good, the human. Like, we are very limited. We have this pair of eyes. We see a limited number of colors, so maybe we're not too good. The best human is very good compared to the worst human, but it's still a human. And with the potential of the AI, we could surpass so much more the human if we have the right molecular validation tools, for instance.
Oh,
It's
yes,
just,
yes.
yeah.
100%. So I can probably sneak peek a little bit. We are just testing on 100,000 samples that are, let's say, 100% accurate. And I can probably share some results later on, but that was the take, because the hypothesis is everything we think is incorrect, as everything we think as a humans is incorrect. Let's check if the AI will discover something we think it's not there. And I mean, I did some tests on this for it's completely different topic, but connected, because I work on the individual identification of animals, right? So it's like naming the animals. And especially this was a case on lynxes, right? So you have lynxes, and the expert, who is someone who is studying and reintroducing the lynxes into Central Europe for 20 years, told me there is no evidence on patterns being transferred from parents to the child, right? And I just run some couple tests, and I discovered that, I mean, there is some way that you can predict with pretty bad accuracy that this is the parent, and it's more than random. So it's I don't know, 70% accurate that you identify the correct parents based on the visual perception. But it's super hard for people to understand why. And since I don't have the answer to support the prediction, it's super complicated to even advocate for this is correct. But I think it tells you the story into, I mean, it tells a good story, because it's yes, you can run some training and some prediction and see, yeah, there is some pattern, but you cannot describe the pattern to humans. So it's actually super complicated, and philosophically and psychologically, it's so complicated. It's so complicated. But interesting, for sure. Very good questions. Probably we could spend a couple hours over a beer to discuss that.
Sounds good. Are you going to be in Davos?
Nope.
Okay.
I'm in US and cannot leave the country, or they will revoke my visa. So.
Oh, okay. Maybe some other time. Yeah, but just to conclude, even if we don't, even if we are not able to use those AI products or outputs to teach humans new things that humans didn't know before, maybe we can incorporate it in some technology, in some, machine, in some commercial product that identifies the species in a cheaper way than the current technology. If we, through images, we can do it cheaper than DNA and molecular, it has an application.
Yes. And I think we are going there with the study I mentioned, with a lot of, sample data. And the question is, is there some measured gain of using the AI to actually get something or not? And if there is, there could be even really a potential for monetizing that at the end. But I'm a little bit skeptical in general. Despite I'm not supposed to be skeptical, but I'm definitely skeptical.
All right, thank you very much.
You're welcome.
So I check whether we have more questions in the chat, but I can't see. And I remember last time we had questions in weird place, but I don't have this sort of thing visible now. So if there is any more question, it's the last moment I would say to ask. If not, then I think I can say again, thank you very much, Lukáš, for this very interesting talk. I'm actually glad that it's recorded because I was contacted by many people that couldn't join today, so they will be happy to have the access and listen to it. And probably some people will contact you with some questions as well. And maybe after some time rethinking different things we've learned today. Yeah, so I think from my side, it's big thanks and for your time and for presenting this all this complex, difficult work for us. And yeah, I don't know, Glenn, if you want to add something, but if there is no more questions, then yeah, we can slowly close the meeting.
I think we can almost close it off. Let me also thank you, Lukáš, for showing how we can work together with these tools and how we can integrate them. It was really fascinating. Not only on the fungi, but also on the plants. I think we can really work with that because plant diversity and fungal diversity are, of course, also somehow linked. And even within FunDive, we have a lot of plots, we have a lot of observations, which will feed into your work, but then your work can also feed into ours. Like just thinking, be nice for our eDNA sampling plots to look at how each of those plots has which is the predicted plant species richness based on this PlantNet data, which I didn't really know about. So it's quite nice. It will have a big impact on mycology because we are such a rather young science with a lot of non-experts, as you said, and it really democratizes the access to these really, we should say there's a lot of species and a lot of difficult species. So in that way, it does democratize the access or it lessens the learning curve for a lot of people to get into mycology, which I do really appreciate. And I might just end with one story from Belgium, is where we had the identification. I was using OPS-Identify, I guess, which was based on some training data from France, and then it seemed that a species that we almost never identified, Sterium insignitum, was present in Belgium. And we only discovered it because it was identified using these apps, which were trained on another country's data. So this is quite nice. And I'll link the article, which is in Dutch, but you can auto-translate it by ChatGPT by now in the chat for the people who are interested in that.
Awesome. And thank you for having me and the kind words. It's always challenging to speak to a different community with different language, and it's hard to balance the technical details and the jargon to use, and it's usually super hard for me to express myself so it's understandable to a general communities or completely different domain experts, but one thing that I laugh is that I think if you are, Glenn, the only one who has those thoughts, I actually did it the right way, because I try to influence the future mycologists to not be afraid of the tools and see some potential how it works in different, different fields. And this aspect of how different plant species and fungi species co-occur and so on is super interesting aspect that is worth to, I mean, it is studied, but it's worth to study it on a longer scale and a bigger scale and longer period and so on. So great that I kind of inspired at least the talk and, future thoughts for that. So thanks a lot for saying that.
Cool. So let's finish it here then. I wish everybody a good night. And if you do have any more questions for Lukáš, you can find all of his info on the FunDive website. This meeting was recorded and we'll put it on YouTube later this week.
We have
Yes,
one
and
raised hand, by the way.
yeah,
Yes, Vladimir, you
Yes.
can go ahead if you want to. You can unmute yourself if you have a question.
I can see you are unmuted, but I cannot hear a thing.
maybe it was not a raised hand, but the clapping and like these two icons are like next to each other. but
Yes, this has happened to me
maybe it could
quite
be-
often as well.
yeah.
Yeah.
So yeah, I can also say thank you very much again, and we see each other in next month, in the last Tuesday of next month, and we will have slime molds as a main topic of the meeting. So stay tuned. We will spread the message in advance. So thank you very much. Thanks again, and have a good night.
Bye, everyone.
Bye,
Have a
everyone.
good one.